Hello, second year. Welcome to our biochemistry laboratory experiment number 2, which is entitled Breaking Proteins. So, for our lecture part, we were able to discuss the basic principles of your proteins and your amino acids as well. So, this will be an application of of what we have learned on your lecture part. This is going to be a laboratory experiment at home. So these are the materials that we will be needing for the laboratory experiment at home number 2. So number 1, we will be needing a stove or microwave. Number 2, pot or microwave safe container to boil water. So it is recommended to use a cup glass, okay, instead of the plastic one because we will be subjecting our glasses into heat. So kapag nag-boil tayo ng water sa ating stove or pag pinasok natin yung ating mga cup sa ating microwave, it is safer to use the glass compared sa plastic kasi may tendency na matunaw yung plastic cups natin. And then, what else? We will be needing a fork. So, it is safer to use yung mga fork na ginagamit natin pag kumakain tayo sa bahay instead yung mga plastic. So, do not worry, ang mga pinaka-reagents or materials na gagamitin natin for our laboratory experiment at home is water and egg lang naman. So, it is easier to use and then mas madali siyang hugasan afterwards. So, wala naman tayong gagamitin na mga toxic materials. Egg and water lang naman. So, safe pa din naman gamitin yung mga glasses natin afterwards. Basta, ugasan lang natin na maigi. Next, we will be needing a pair of scissors, 1 bowl, and 4 small glass containers of the same size. Again, 4 small glass. Okay, glass po ang kailangan natin kasi nga, we will be subjecting our glasses or our cups into heat. So, dapat glass ang ating gagamitin. Containers of the same size. So, hindi naman po kailangan na exact same size sila. At least, magkakalapit yung sizes nila. Hindi malaki yung difference. Next, 1 egg that will be split into 4 parts. And then, 2/3 cup of water, which has a 150 milliliters of the volume, 1/3 cup of your rubbing alcohol or 75 ml, and then you will be needing a tape and a pen for the labeling. So if you're going to ask me kung paano kayo measure ng 150 ml and then 75 ml, hindi naman po kailangan accurate na 150 ml kasi wala naman tayong mga biret and then mga graduated cylinder sa bahay. So as medtech na smart medtech, a wise medtech, pwede po tayong gumamit ng syringe or kung meron kayong known volume ng mga container sa bahay, kung meron kayo Nature Spring na 200 ml, bawasan lang ng 50, mag-aspirate lang ng 50 ml ng syringe, and then, yun na, makakakuha ka na ng 150 ml. So, kayang-kaya nyo na po mag-measure niyan sa bahay. Okay, pwede pong gumamit ng syringe, pwede din tayong gumamit ng known volume na container. For example, yung mga Nature Spring natin sa bahay. And then, this is the step number 1 for our laboratory experiment at home. Number 1, pour 1/3 cup of rubbing alcohol into 1 glass container. Room temperature water, 1/3 cup into another, and then the rest of 1/3 cup into a microwave safe container or into a pot. Do not forget to label your glasses. So, ang magiging output nyo dyan, at least meron kayong 4 glasses. And then you have to label all of them. Number 1, control, tap water, alcohol, and then hot water. So gumamit lang po yung ating scientist dito, yung nagde-demo natin ng isang cup, literal na 1/3 cup. So kung may pang baking cup kayo, and then 1/3 cup naman yun, so wala naman pong problema. And then crack the egg into the bowl, into a separate bowl, and then remove the yolk. Please, please, please remove the yolk. Number 3, cut the egg white into pieces. So, you can add 1/4 into each glass container. So, gamit ang ating gunting, ang ating pair of scissors, you have to cut your egg white into 1/4. So, at least 4 siya. And then, heat up the water for your hot water treatment and pour into one of the empty glasses. Caution, please be careful on handling the hot or the boiling water. Next, quickly put 1/4 of the egg white into the boiling water. Then put 1/4 into the alcohol, 1/4 into the room temperature water, and the remainder into the final empty glass container. So, nung hinahati nyo na yung inyong egg white into 4, ilalagay nyo na siya doon sa inyong mga container, sa inyong mga glasses. So, 4 yung na-separate nyo, 4 din yung paglalagyan nyo na baso. And then observe any immediate changes that occur in the terms of egg white color and consistency. If you try stirring the different treatments, rinse your fork between stirs. So advisable dito na kung mag-steer kayo, i-rinse nyo yung inyong fork every after yung gagamitin. Kunwari, si hot water muna. Inano mo siya, ni-steer mo siya. So, babanlawan mo muna yung inyong fork and then tsaka mo siya isa-subject dito sa alcohol para hindi po magkaroon ng cross-contamination between these 4 glass containers. Or kung meron naman kayong 4 na fork sa bahay, at least, di ba, tagi-tagisang fork na lang sila para hindi ka na mahirapan maghugas. So, kung tatanungin po, ma'am, dapat po bang distill, dapat po bang alkaline water or tap water, wala naman pong problema kahit ano. And then wait for 30 minutes. Use the fork to inspect the state of the egg whites in each treatment and know how they may have changed over time. So the principle behind our laboratory experiment home is what we call the denaturation by heat. Heat can be used to disrupt hydrogen bonds in nonpolar hydrophobic interactions. These occur because heat increases the kinetic energy and causes the molecules to vibrate so rapidly and violently that the bonds are disrupted. Gentle heating causes a reversible denaturation of protein, and then vigorous heating causes irreversible denaturation of protein. And then lastly, heat coagulates and destroys protein present in the bacteria. Thus, sterilization of instruments used in medical settings requires the high temperatures. So that's it for our laboratory experiment at home. I'll be providing YouTube video, parang guide niyo siya on how to do your laboratory experiment at home. So after niyo pong gawin ng inyong experiment, of course, you will be answering your worksheet. Okay? Ito ay isang 50-point worksheet. So you have to answer and then attach your pictures or your videos here. So kung hindi kasya, pwede natin siyang i-attach on our Google Classroom. Kindly attach the Google Drive link for your experiment at home. So that concludes our pre-lab discussion for your breaking of the protein. So good luck, everyone, and God bless. Thank you.